Hi all, welcome to the Physiology study series. Today we will see what is the criteria for a blood smear to be called as an ideal blood smear or simply ideal peripheral blood smear. An ideal peripheral blood smear, it should be salmon pink in appearance. It should not be too violet or too pale in appearance. Not only that, and the smear should occupy at least 3/4 of the slide. Smear should be tongue shaped. And when we do the peripheral blood smear, it should have a head, it should have a body, and it should have a tail. And in a peripheral blood smear, all the WBC, they are unevenly distributed. On the margins, you will find monocytes and polymorphs, and even at the tail. And in the body of the film, you will see lot of lymphocytes. This uneven distribution of cells is a normal finding in a peripheral blood smear. And the smear that we take, it should have or it should be one cell in thickness. That is, a well-made film is one cell in thickness. However, one cell in thickness peripheral blood smear looks like in an oil immersion lens. So this is a peripheral blood smear that has been kept under oil immersion lens. Here we are able to see RBC. These RBCs are they are evenly spaced, and only few RBCs they are overlapping on one another, and rest of them are evenly distributed in singly. This slide, which has a blood film, which is called a thick peripheral blood smear, and this is not an ideal blood smear. We'll see in the next slide how under oil immersion objective, how this thick smear looks. This is how a peripheral blood smear which is too thick appears under oil immersion objective. Here you are able to see different RBC, many RBC which are occurring in groups. They are not single and they are not evenly spaced. They are in aggregations or they are in groups. So this will occur when a smear that has been taken which is too thick. If you move the spreader too fast or too quick on the glass slide, it will produce a too thin film. How you can identify different cells or how you can identify a smear that is too thin? Here under oil immersion lens, a thin film appears like this, or you are able to see all the RBC, they have lost central pallor. So that is one of the findings where we can say that a smear which is too thin, and it is giving a false impression of spherocytosis. This slide is also an example of not an ideal smear because it's an irregularly patchy film, and the smear is prepared on a dusty slide. And here you are able to see many gaps. This blood smear which is prepared on the glass slide is also not an ideal smear because it has not a single tail but many tails. So this is a film that has been spread with inconsistent pressure and using an irregularly edged pressure, resulting in long tails. This smear is also not an example of an ideal blood smear because it has many air gaps and vacuoles in the smear. How come a smear will appear like this? When you take a glass slide which is not thoroughly washed or cleaned properly, it will result in this very greasy slide. So we have seen in this particular video the criteria of an ideal peripheral blood smear. Thank you all.